Essentially, there are 7 steps to making a direwolf. The first step is to find actual direwolf fossils. We go out into the field and to museums and we find remains that look like they're going to be really well preserved. Second, we have to generate this genome sequence from a direwolf. So we use the tools of ancient DNA. We take the fossils that we found, we grind them up, and we extract preserved DNA. That DNA is not going to be in great condition, but we can still get all of the information that we need, even if the tiny little DNA fragments are broken and damaged and really small. The third step, then, is to identify what it is that made a direwolf like a direwolf and not like a gray wolf. To do this, we line the direwolf genome up against the gray wolf genome on a computer, and we scan them, looking for where they're different from each other. This is how we identify the core genes. What is it in that DNA code that makes a direwolf a direwolf? Once we've identified the genes that we want to change, it's time to design the tools that we're going to use to actually make those edits in living cells. This is part of the genome engineering process. After we've done that, we move on to step 5, which is actually using those tools to make the edits. This is where we're working with cells in a dish in a lab, and we are actually going into the genomes of those cells and cutting out the gray wolf versions of the genes and pasting in their place the direwolf versions of those genes. After we have those edited cells, it's time to turn that into a living animal. So step 6, embryo transfer. This is where we take these cells and we take another cell that has had its nucleus removed, so it doesn't have any of its own DNA, and we inject in its place our edited genome sequence. And then we're on to the final step. Transfer those edited embryos into a surrogate maternal host who's going to eventually give birth to our direwolf.